Hello everybody, this is my CRISPR presentation made by yours truly, Andrew Fairweather. Let's get going. What is CRISPR? Clustered regular interspaced short palindromic repeats, or better known as CRISPR, is a means of genetically modifying DNA, or in another term, genomic editing. It was originally a nuclease called Cas9, which was altered so that instead of just targeting viral DNA strands, it could be applicable to any cell's DNA for modification. CRISPR can alter and change certain traits within DNA and in an organism. Such traits can be amplified, like increasing a wood's pulp and fiber yield by editing the code of the DNA to desired traits. CRISPR is also used as a way to identify viruses along with how they function so that treatments can be procured for said viruses. What is Cas9? Cas9 is a nuclease that was altered genetically, originating from bacteria cells which would use Cas9 as an immune response to bacteriophages' DNA strand attacks. What Cas9 does is it uses guide RNA strands to latch onto the viral DNA strands, which it then cuts it, making it unable to replicate and reproduce within the cell itself, disabling it entirely. I also have photos here showing the process of Cas9 using the guide RNA strand to immobilize the viral DNA, allowing for it to be cut. What areas is CRISPR best utilized? CRISPR has many fields where it can be applicable, such as editing the genomic coding between mosquitoes so that vector-borne diseases are harder to pass on from victim to victim as they drink more blood. Creating disease and climate-resistant crops are also another, so that they say you want to have a higher yield of corn, but you're having a lot of problems like with the weather. In that case, you use CRISPR to alter the genes of the corn so that it is better resistant against the weather, advancing medicine and human health. Things such as finding a better way to cure certain diseases, for example sickle cell, cystic fibrosis, and cancer treatments, and using CRISPR to edit the genomic strands within those set diseases and mutations to find the cures for them. Lastly, livestock and the health of animals. An example would be the methane output that cows have. We're able to use CRISPR to limit that and actually decrease the amount of methane they produce from eating so much grass, along with the treatment with disease to produce healthier offspring. As you can see here, a little table, and the next one. What can CRISPR do to help people? The ways that CRISPR can help people are the altering of animal organs for xenotransplantation, where say a person has severe third-degree burns all over their right arm. They can be aided by having altered pig skin that is edited, which is then grafted onto the person's burnt arm. This way their skin can heal, replacing the burnt off and charred skin. Another is the implantation of healthier cells to replace and cure sickly cells. This is where you take a cell that is sickly or mutated and edit it with the correct strand using CRISPR. You then put it back into the body once the cells have multiplied enough. This way the mutation is slowly fixed by the body recognizing the newly altered cells. What achievements has CRISPR achieved? CRISPR, I'd say, has made two major breakthroughs, being the sickle cell treatment and cancer treatment. For the sickle cell treatment, the way that it works is they take hemoglobin outside the body, and then they use CRISPR to edit the genome using CRISPR-Cas9, and then cuts out the regulatory gene, making it so that fetal hemoglobin starts being produced a lot again. Once there has been enough fetal hemoglobin produced, it is then infused back into the body, and once that is done, the mutated sickle cells will become healed and they're turned into normal red blood cells. For the cancer treatment, the way it is done is doctors extract T cells from the patient. CRISPR uses editing to change the DNA to better recognize tumor cells and to also remove the breaks that limit immune response. Currently, I haven't gone so far in clinical trials such as leukemia and lymphoma, but researchers have shown that genes like PD-1 that inhibit immune responses to tumors can be edited out. What are the limitations of CRISPR? Since I've talked so much about the good things, let's talk about the bad. Many of the limitations of CRISPR are that the protein Cas9 is enlarged, so when it is to be placed into cells, it is difficult to insert it. And because of that, the immune response may occur with a failed insertion, where the immune system can view the Cas9 protein as a possible threat and attack it, making it even harder for the cell and DNA alteration to work. Another is accidental genomic editing, which occurs when strands of DNA that weren't supposed to be edited are snipped and get snipped by the Cas9 protein, which is only a problem that will be fixed with time and technological advancements. Currently, CRISPR is only viable for single cell gene mutations, meaning diseases just such as heart disease, diabetes, and most cancer diseases, which are more complex genomes, are untreatable because they're too complex. Mosaicism may also occur where the cell is not edited properly and causes coding errors within the DNA that may lead to possible problems with the treatment. The treatments are also not permanent. Even if you do get a CRISPR treatment on certain cells and you want to bring out certain traces in the DNA, the effects may not be permanent and you may have to restart editing. Lastly, CRISPR treatments are extremely expensive themselves, which they can cost up to around $1 million per person per treatment, making treatments very hard to get and very limited for the average person. What are the ethical concerns with CRISPR? The ethical side for animals is that experimentation should not be done point blank. This is often because people view animal experimentation as some form of chemical torture, where they're putting acid or sharp objects into the animal, when in reality for CRISPR, it's more so taking cells outside of the animals painlessly, editing them, and then inserting them back into them to see the reaction that may occur. For humans, it's just seen as inhuman and goes against human rights. This is because of warped perception from media and cinema, where human experimentation is seen as this vile thing, along with the horrible human experiments in the past being used as a stance to deny any notion of possible experimentation. This ties into the final ethical point, being that scientific progress should not come at the cost of others, in the sense that either their livelihood or possible loss of life from said experiment. My conclusion overall for this is CRISPR has provided many benefits to humanity and has aided in technological and pharmaceutical progress. However, it is unlikely we'll see any massive breakthroughs in the field for the next couple of decades, such as cures for cancer entirely, all types of cancer, bone diseases, joint diseases, things such as that. They probably won't be cured and fixed for a long time until technology gets to a high enough point where they can be. These are my references. They're actually quite interesting. I'll link them in my post. And thank you all very much for listening. You guys have a great day. Au revoir.